como tal el seminario, pero nos acompaña la profesora Paola Pulido, quien amablemente invitó a nuestro último conferencista, digamos que esta es la sesión de cierre, entonces es una sesión especial, digámoslo así. El registro de asistencia, vamos a pasar dos listas, una para los estudiantes de la profesora Carolina, entonces por favor, ahí lo voy a anotar para que firmen esa, y la otra si pueden hacerlo sin problema. La metodología del seminario es, tenemos la intervención de más o menos 40-45 minutos y al final pueden hacer las preguntas, si es importante que estemos, que me digan para pasarles el micrófono porque si no la grabación no va a quedar. Y las pueden hacer en español o en inglés, como ustedes quieran. Si la quieren hacer en español, la profesora Paola les ayuda con la traducción y también se pueden conectar por Zoom, si quieren les envío el enlace, o las pueden colocar en inglés también por Zoom. ¿Listo? Entonces, pues sin más preámbulo, muchísimas gracias, profesora Paola, y pues bienvenido.
Gracias, Vero, muchas gracias. Hola a todos, muy buenos días. Los que también son de mi curso, la hoja es amarilla, o sea, no hay pierde, ¿ok? Pues para mí es un gusto tener la oportunidad de presentar a Kevin hoy en este último seminario y voy a hacer la presentación en inglés porque todo el seminario pues va a ser en inglés, ¿vale? So I'm super happy to be here and with you, and I'm thrilled to have you at Universidad del Rosario. Thank you for being here. So Kevin did his bachelor and master studies at the University of Toronto in Canada. And that's where I met him. I think it was already like 10 years ago when he was doing his master's and I was doing my PhD. So he was at the museum, right, with Robert Murphy, and I was a different campus but at the same university. So we had the chance to share different ideas, talk about phylogenetic methods, bioinformatics, and it was a very good experience. Right after that, Kevin went to work with Professor Laura Kutbatko, right? And Laura is based at the Ohio State University, and that's where he did his PhD in evolution, ecology, and organism biology. And then he did a postdoctoral fellowship at the Institute for Discovery at the University of Wisconsin in Madison, right? At the Solis Lemus Lab. So people that have worked with hybridization and speciation might have already seen some papers from that lab. It's super interesting, and he might be talking about that in a bit. And also, right now, he's working at the Institute for Computational and Experimental Research in Mathematics at Brown University. So Kevin is interested in developing and understanding methods for inferring phylogenetic networks and hybrid speciation using genomic data under the coalescent model. And so this is the title of his seminar. Thank you for being here.
Yeah, thank you very much, Dr. Pulido. I'm very excited to be in Colombia. So as Professor Pulido said, we met 10 years ago and we spent many days discussing about scientific ideas, and Colombia was always on top of my list to visit around the globe. And this is my first country in South America to visit. Unfortunately, I don't speak Spanish, but I really enjoyed a lot of food. I really enjoyed great people in Colombia. I saw many great animals and plants. And it's been 2 weeks so far, so time flies, and I'm leaving tomorrow morning. But I just want to say thank you, Colombia, for having me and letting me have a great time. All right, so I'm gonna talk about some stuff about detecting hybridization and also inferring what's called phylogenetic network that illustrates both speciation and hybridization events on a phylogeny using genomic data. So this is a artificial intelligent image created by AI. I used DALL-E, and I typed in, okay, DALL-E, give me an AI image that says a phylogenetic tree and show me genomic data and show me a bunch of animals. And this is a picture that it gave me. And I think this really captures the kind of research that I'm doing. So we see a tree that is going away from the earth and is somewhat bifurcating. Sometimes it's not. So it can be either a bifurcating tree or a network. We see a bunch of species, animal and plant species, starting from dinosaurs, mammals, and some fishes and stuff. And they're all surrounded by this big, huge genome data. And we're going towards the sun, which is try to understand and try to see the whole picture of a phylogeny that describes the relationship among these species. So everybody's familiar with the phylogeny, right? I hope, yep. So starting from the very basics, let's start from the very basics. This is a cartoon example of a phylogenetic tree that contains 5 species, and they're labeled on the bottom A, B, C, D, and E. And on top, we have a root, which is a single node that represents the most recent common ancestor of all of the species under consideration and all of the internal nodes. And in between the root and the tips, we have a series of internal tree nodes where a single branch splits into 2, denoting a speciation event, which we can explain biologically, a single ancestor species giving rise to 2 descendant species. So this phylogenetic tree has been extremely useful in practice. And for example, a tree of life, which is a single phylogeny that shows the evolutionary relationship of all of the organisms that have existed or are existing on our planet. And this basically actually shows what kind of biological processes have shaped the biodiversity that we observe today and how different species are related with one another. And also, besides only understanding how species are related, they're also useful in forensic, for example, or medical science, conservation science, and many more. And there are phylogenies are used in linguistics to understand how different languages are related with one another. And there are some examples of phylogenies of music genres to understand how different music genres evolve. So phylogenetic trees has been extremely useful. However, the question is a tree a perfect model to depict all of the biological processes that have shaped what we observe today? Unfortunately, no, because phylogenetic trees assumes a novel species can only arise via a speciation event. But in practice and in nature, hybridization is much more common. Historically, hybridization was seen as something unnatural, and even if it somehow happens, it's not going to persist over evolutionary time. So in biological study, we can kind of ignore hybridization. That was what happened decades ago. But thanks to genomic data, we now know hybridization is actually much common than we previously thought. And there are a bunch of examples, like this mule here, which is a hybrid between female horse and male donkey. Heliconius butterflies, which a lot of research was done in this here in Colombia. And hybridization is seen quite apparent in the wing pattern here. So for example, a hybrid butterfly will have a wing pattern that is somewhat intermediate between 2 parental species. And also birds. Birds are very well known for their rampant occurrence of hybridization. So one way to deal with this hybridization for phylogenetic studies, we simply ignore their presence. We're not gonna care about hybridization. We just conduct phylogenetic analysis using trees and hoping we're gonna get the true answer. But that's not a good practice, because no matter how much data you have, no matter how good computer you have, no matter how long you run your analysis, you are going to get what's called biased species tree estimation, which means the tree that you get is not going to reflect the true relationship between species. So as an alternative, a concept of phylogenetic network was proposed. So network is basically a generalization of the trees by allowing 2 branches that are once separated to merge at a single node, creating a structure called reticulation, which is shown in this pink triangle here. So this basically represents more complex evolutionary history than speciation by showing hybridization events. So in this case, we can say species D is the hybrid species between species C and species E, and so on. So this is a kind of phylogeny that I'm going to talk about for the rest of my talk. And we're gonna stick to what's called binary level 1 rooted explicit networks. I'm going to talk about what I mean by these terms, but binary means at every tree node, a branch splits into two. So at every tree node, two descendant species will happen. And every reticulation node, there will be always two parental species giving rise to one hybrid offspring, which can subsequently divide into multiple species. And level 1 network means relatively simple network, rooted, which means it has directionality starting from the root towards the leaves, and explicit networks telling us the evolutionary history using this phylogenetic network. But in practice, especially in biological science, when they say, oh, we have conducted a network analysis using our data, in many cases, they mean they computed something like this. This is what's called the abstract network. And this very complex structure basically shows how different sequences are superficially similar to each other, but not necessarily telling us how they're evolutionarily related. And they're very complex in their structure. So it tells us the data has somewhat complex relationship, but it is really difficult to depict what's actually going on in these small cycles or small reticulations here. On the other hand, I'm going to talk about explicit networks. An example of explicit network is here. It tells us the evolutionary relationship between species. So here's an example that I stole from Fontaine et al., published in Science. Here is the phylogenetic network of 6 mosquito species that occurs in Asia. And the black line represents the tree or tree branches. And the green lines that are directed towards the one branch to another represents a reticulation or hybridization or gene flow event. So by here, we can see we have a bidirectional gene flow between this yellow and pink distribution of the mosquito species. And here we have a unidirectional gene flow from the green towards the purple species distribution. So explicit networks actually tells us something more about biology. So to make it more nutshell summary, abstract networks are good because they result in colorful and intuitive diagrams that shows how data are related. They're very fast to compute because they use what's called distance-based metric, which basically counts the number of differences on their, between sequences. They're unrooted, which means we can't understand the directionality evolution, which probably is the most basic information that we want to extract from any phylogenetic investigation, because we want to understand the ancestor-descendant relationships between taxa. So the goal of this abstract network is to display conflicting signals in the data, but not necessarily to depict evolutionary relationships. On the other hand, explicit network resembles much similar to phylogenetic trees, and they're much smaller than the abstract networks because they're very computationally intensive to estimate. And when I say computationally intensive, it's going to take days and weeks to complete the analysis. And they're either rooted, so it has directionality from root to tips, or semi-directed, which is an unrooted network that does not really tell us the relationship among the tree branches, but it tells us who are the hybrid parents and who are the hybrid offsprings, and it infers complex evolutionary histories. So it's pretty clear that explicit networks are the one that is going to be useful in biology. Abstract networks are the ones that is useful to display the conflicting signals in the data. But unfortunately, that's not what's actually happening in practice. So this is a paper that was published in the beginning of the COVID-19 pandemic. And I was in America doing my PhD, and the lockdown just began. I was stuck at my home, just hoping that I don't get COVID because I was really scared. I thought I will die if I get COVID, so I was only staying at home. And we found this paper that has a title, a very ambitious title. It says, Phylogenetic Network Analysis of COVID-19 Genomes. And in this paper, they had a single figure, which is a network that is shown here. This is an example of abstract network called median joining network. And this is great that they're trying to do something with COVID-19 genomes when we literally didn't know anything. But the problem is they start to understand the origin of pandemic using this network. And they also try to understand the transmission history of virus only based on this based on this network. So myself, along with a couple of Colombian biologists. We wrote a paper to the PNSA paper. And then here we argued, and we had a very strong title saying, median joining network of COVID-19 genomes is neither phylogenetic nor evolutionary. And here we said, because median joining network is unrooted, we can't understand the directionality evolution. If this is true, how can we understand where it starts and which direction it's going? The second point is, even though somehow this median joining network tells us the true relationship between genomes, it's not really good practice to trace the entire history, transmission history of infection based on a single phylogeny. And there were some, after we submitted this paper, there were some rebutters. We got a couple emails from the original authors. You guys are wrong. And then we wanted to say, actually, you guys are wrong, and back and forth. But it eventually faded out. So what should have they done? They should have inferred explicit networks. So in the past 20 years or so, computer scientists, mathematicians, biologists, statisticians, they have been working together very hard to understand the structural properties of explicit networks. And they have proposed multiple subclasses of these explicit networks, and now we have more than 20 subclasses. And this diagram, which is published in this review paper that we wrote, represents the relationship between different subclasses of explicit networks. So each of the nodes represents different subclasses, and they're linked by an edge based on their structural similarity. Unfortunately, many of the cases, they are not really useful for biological study because they were too complicated. Actually, but only few of them were biologically relevant. For example, this K-nested network, which is very pretty and which looks quite complicated, but unfortunately it's not biologically significant because it's difficult to make biological story out of it, but more importantly, it's difficult to infer this from the data. So one important subclasses of explicit networks that are useful in biology are the trees. And tree here, we have a trees on the bottom. Tree is basically one condition of explicit network where the structural property is lack of reticulation. So another subclass of network that are very useful in practice is this level 1 network, which is very closely related with the trees. So here I show an example of level 1 network. So the structural property of level 1 network is it has reticulation that is absent in trees, but each of these reticulations are isolated from each other, which can be biologically interpreted as hybridization event will always occur isolated or independent from one another. So many of the computational methods that can infer networks from genomic data are assuming the true network lies in this class of level 1 networks, but currently we are going forward from inferring only level 1 networks towards level 2 networks. So level 2 networks assumes, okay, you can have multiple reticulations in the same blob, which is a big circle that is composed of reticulation, but you can have maximum 2 reticulations. And sometimes you can have another reticulation branch, and then you can have a blob or a cluster of reticulation that have 3 reticulations. It's going to call level 3 networks. So this level of networks basically quantifies the interwovenness of the networks. Anyway, but for the rest of the talk, we're going to stick with this level 1 networks because that's what we can infer from genomic data. So we have a handful of computational methods that can infer networks from genomic data, and they can largely classify it into summary or full data methods. And this is a small table that are popularly used methods that infers explicit networks. So summary methods basically, as name suggests, it summarizes genomic sequence data into some other form, and in many cases is gene trees. So they define the delimitation of each gene, and they try to infer the history of the genes, each of the genes, and they use those gene trees as input to infer the networks, which makes them to be relatively fast to estimate because a lot of hard work has been done prior to the network analysis. But the biggest downside is that their accuracy completely depends on the quality of the estimated input gene trees, not the sequence data itself, which can be very erroneous. On the other hand, full data models uses sequence data as input because it fully models the evolution of sequences under the network. But they're computationally very intensive to estimate. And when I say intensive to estimate, if you have 5 species or 5 sequences, it's going to take days to finish the analysis. So they are not really computationally feasible. So as an alternative to this network analysis, what people are often doing is what's called hybrid detection method. So these hybrid detection methods are largely categorized into population clustering methods, and we have 2 very popular methods called structure and admixture, which often give us a plot that looks something like this that visualizes the result, the analysis result. On the other hand, we also have what's called site pattern frequency-based method, and usually 2 popular methods there that's being used in practice are HIDE and the D statistics, or also called the ABABABA test. So the basic idea of this site pattern frequency data method is it tries to see 4 taxa at a time, and it's trying to see a site pattern that is conflicting with the species tree that is shown here. And when these 2 patterns that are conflicting with the species tree occurs in the similar frequency, okay, this is probably, hybridization is not the main cause of this occurrence of these conflicting site patterns, but it probably is what's called incomplete lineage sorting, which can result in very similar footprint in organism's genome. So going into a little bit more detail, we've been working a lot to understand the limitations and advantages of these popular hybrid detection methods. So focusing on population clustering methods first, the good advantage of these methods is they can handle large data set with many individuals. And this is possible because their primary goal is to understand how different individuals can be clustered with each other so we can delimitate different populations. So you can deal with tens or hundreds of sequences at a time. But identifying hybrids involves subjectivity because identifying hybrids hybrids are identified using this plot here. And so each of the color basically represents the probability that an individual will belong to a specific population. So more specifically, each of the line, the top to bottom line represents an individual, and each line will have 1 color or 2 colors. And when there are 2 colors, this individual has probability of belonging to one population and another probability of belonging to another population. And when this probability is about half, as shown in some individual here, they basically define, oh, okay, this is a hybrid individual. But this is a subjective process because, what about this individual? Like it's still half and half, little less than half. Is it a hybrid or is it not a hybrid? It's really difficult to draw a line. So that's the biggest problem of using population clustering method to detect hybrids. And admixture, which uses maximum likelihood framework, often fails to detect hybrids. So even though you have a good data and you run admixture analysis, it's going to tell you there is no hybridization here when there actually is hybridization. And structure, which is more popular than admixture, the structure can actually detect hybrid individuals more frequently than in admixture, but it can't distinguish signals from hybridization and incomplete lineage sorting, which means if it's a tree, if a population or a bunch of species experience adaptive radiation, it's going to tell us there is hybridization when there is no hybridization. And they often result in inaccurate estimate of ancestry coefficient, which basically is used to draw these figures. So you can't really trust the proportion of these colors using structure. But it sometimes detects recent hybridization when both parents that played a role in hybridization and hybrid offspring exist, but only recent ones. And it's unable to detect what's called ghost introgression, which is a case where one of the parents went extinct or unsampled, so it doesn't exist in the data. So it completely fails to detect that. And it makes sense because we don't have data, we can't see hybridization. On the other hand, site pattern frequency-based methods, the detection power outperforms population clustering methods in most of the cases. And they can actually distinguish the signals from hybridization and incomplete lineage sorting when large amount of data is given. And Heidi, which actually estimates what's called inheritance probability, that is very similar to ancestral coefficient. I'm going to talk about what I mean by inheritance probability in a minute. They have very high accuracy in estimating this parameter. But some of the recent studies in Frankel and Anne and Pang and Zhang, they showed these methods, although they kind of working well in some conditions, they have very high false positive rates in the presence of rate variation, which means when different species, the rate of mutation is different among different tree branches or hybrid branches, they're not going to do a very good job. And also, even though these methods can detect ghost introgression, it can differentiate it from hybridization among extant species. What I mean is, if one of the species went extinct or unsampled, it's going to still tell us there is a hybridization, even though one of the parents is absent in the data. But we can't really distinguish, is this due to ghost introgression, or is this because there is a parent exists in the data? And finally, the ABA-BABA test requires known species relationships. So we need to locate each of the species precisely on the right place on the topology, and if we mix up the position of this taxa, it's going to tell us wrong answer. So you need to be very cautious, even though they work quite well, when you're using site pattern frequency-based methods to detect hybrids. And these hybrid detection methods are slowly disappearing because we have very rapid development of network methods. But still, we are still working on this hybrid detection methods because we are really excited about this method. So here I introduced two papers that are from our lab. So the top paper is a new method to detect hybridization from genomic data. And this is similar to ABA-BABA test and Heidi. In a way, it uses site pattern frequency, but we use larger amount of data to detect hybrids. And this was actually published yesterday. So before I make the slide today, it was a preprint, but I captured it for the paper. Here we show an example of the performance of our method. Here, the green line represents Heidi, red line represents the ABA-BABA test, and blue line represents our methods proposed in this top paper. X-axis represents different conditions of hybridization. Y-axis represents the detection power. As you can see, as we increase the proportion of genomic contribution from one parent towards the hybrid, the power of our method clearly outperforms Heidi and the ABA-BABA test, which was already a pretty good method. And on the bottom paper, it basically shows what it talks about is we can actually use the outputs of these hybrid detection methods from ABA-BABA test or Heidi, which actually conducts statistical tests and give us p-value. We can basically merge all results from those analyses and tell us if the data in hand is better to be analyzed using trees or the networks. So this probably will be one of the first steps if you want to decide if you want to do tree analysis or the network analysis. Okay, so hybrid detection methods are useful, but they don't really tell us the relationship between different species. They only tell us if there is a hybridization in the data or no hybridization. So the question becomes, this is a main question of my study, can we estimate a phylogenetic network directly from the sequence data efficiently, as efficient as the summary method, which infers gene trees in prior, and accurately, as accurate as the full data method, which utilizes all of the signal in the genome data? And the long answer short, the answer is yes, and hopefully this is why I'm here. And we do so by combining what's called the composite likelihood framework and the site pattern frequency data. I have mentioned site pattern multiple times, but what I mean by composite likelihood framework is because computing the full likelihood of the big topology is computationally heavy. Maybe that's not a good way to go. What we're going to do is we're going to break the big problem into smaller pieces. We're going to compute likelihood on each of those smaller pieces. We're going to combine them to find the answer for the big problem. That's what I mean by composite likelihood. Okay, so site pattern. I have been saying site patterns, quartet site patterns multiple times in the past few minutes, but let's consider this model cartoon example of a network and the corresponding sequence data here. If we look at the very first column of this sequence data, we're going to observe A, and then we're going to place them at the tips of the topology. And this is a site pattern of A. But this site pattern does not really tell us much information. The only thing that tells us is, at the first site of the sequence data, all 5 species shares the same nucleotide. But as we go on the sequence data, sometimes we will see some variation, and we do the same thing by putting them at the tips of the topology. We will get a site pattern of TT-AAT that tells us a bit more story, where this species and this species share the same nucleotide that is different from everybody else. And you might wonder, okay, this data type actually sounds awfully similar to what's called single nucleotide polymorphism, SNP data. But we distinguish our data from the SNP data. We call this data type by looking at the pattern at each site as coalescent independent site. And we assume that each of the site has their own coalescent evolutionary history. So they are different from the SNP data. And the nice thing about this coalescent independent site is we also utilize those sites where no variation was observed, and we also utilize the sites where variation observed, which is also used in SNP data. So this is what I mean by site patterns. So for the next few slides, I'm going to talk about how we can use these site patterns and composite likelihood framework to estimate phylogenetic networks. But before I do that, I'm going to tell you a number of parameters that we are interested in estimating. The first one is tau, that represents a vector of speciation times, where each of the element is assigned to each of the tree node. And this speciation times is measured in coalescent unit, which is a function of population size and the generation times. And they are measured backward in time, which means it is 0 at the tip, and the value increases as you move towards the root. Second parameter is gamma, which denotes inheritance probability that is assigned to one of 2 incoming reticulation edges. And here you can see we have assigned gamma here, this reticulation edge going towards the reticulation node. And because this inheritance probability, the value ranges between 0 and 1, it makes the inheritance probability for the other incoming edge as 1 minus gamma. And biologically, this parameter can be interpreted as the proportion of genomic contribution from one parent towards the hybrid. So when the gamma is 0.5 and 0.5, it means basically about half of the genomic portion was contributed from parent E, another half from parent C, and D will have half of chromosome or half of genomic data from each of the parents. And finally, we have theta that denotes population size, which we assume there is a single population size throughout the entire topology. This is biologically unrealistic, but we assume that for computational tractability. So the first thing we want to do to estimate the likelihood or composite likelihood of the network shown in the previous slide is we're going to break it down into a set of display trees. And what I mean by display tree is a tree that can be obtained by removing one of 2 reticulation edges at a time. So for example, if we remove this reticulation edge, we will get our first display tree where D and E forms a cherry or shares the common ancestor. And when we remove this reticulation edge, we're going to get this display tree where C and D forms shares a common ancestor. And each of this display tree will have some probability that can be obtained from inheritance probability that is assigned to each of the reticulation branch. So more specifically, the probability of each display tree can be computed by multiplying all of the inheritance probability that is assigned to each of the reticulation edge that is not removed in the production of each of the display tree. So display tree 1 will have probability gamma, display tree 2 will have probability of 1 minus gamma. So the second thing we want to do is for each of the display tree, we want to break them further down into a set of quartets. And what I mean by quartet is simply a species tree that has 4 tips. So how do we do it? We select 4 species from our display tree at a time, along with some of the internal tree nodes that represents the most recent common ancestor for every pair of species. For example, if we select species A, B, C, and D from our first display tree, we will get our first quartet that has species A, B, and C, and D, as well as some of the internal tree nodes. And we repeat this process for every combination of 4 taxa, and we can extract 5 quartets from this display tree that has 5 species. And of course, we do the same thing for the other display tree. So at the end, from a single network, we will have 10 quartets that are broken down from the network. And one thing to remember is each of this quartet will have the probability that is same as the display tree that the quartet was decomposed from. Is everything clear so far? Any questions? All right, good. Okay, so next thing we want to do is we want to compute the likelihood of each of these quartets. And given that there are 4 tips at a time, and at a given site, each tip can have any one of 4 nucleotides A, C, T, or G. So there are, how many site patterns are there? There are 4 to the 4, or 256 possible site patterns can arise from a quartet tree. And these 256 site patterns can be represented in a flattened matrix here. And this matrix is 16 by 16, where each of the row represents the sites that are observed in species A and B, 2 species on the right side, and each of the columns represent the sites that exist in species C and D, or the left 2 species in the species quartet. And each of these elements in 16 by 16 matrix will basically tell us the probability of observing site pattern AAAA or ACAA and so on. So the sum of elements in this entire matrix will be equal to 1. So the nice thing is letting I, J, K, and L denotes the distinct nucleotide A, C, T, or G. Under the Jukes canal model, which is a very simple, the simplest molecular evolution model, and the multi-species coalescent, some of the elements will have the equal probability. So what I mean is, given we know the base frequency or the root will have the probability of 25% to be C, A, C, or T, or G, the probability of observing site pattern AAAA or TTTT at the tip of the topology will be equal. So using this idea, okay, some of these elements will be equal, we can further summarize this 16 by 16 matrix by a vector of 15 elements. And this is the 15 possible site patterns that can arise from this 16 by 16 matrix. And the nice thing is, the true probability of each of these 15 elements can be computed given a tree, and given a quartet tree and the taus, which is the speciation times and the population size parameter, which is shown in Schiffman and Kobako 2015. And at this point, you might wonder, okay, Kevin, this sounds all good, but why are you using Jukes canal? You know, this is the simplest model. Like, why are you using it? We know it's not going to, you know, it's not going to always explain what's actually happening in nature. This is way too simple. And the reason is, it's just mathematically too difficult to compute each of the site patterns. And here we show an example of a formula that computes one of the site patterns. And here on the bottom, we have a formula to compute one of the site patterns for symmetric or asymmetric quartet. If the simplest model is this difficult, we are not really trying to make it more complex. Actually, the nice thing is this model kind of robust to model violation. So even though the data is not actually generated under the Jukes scanner, it kind of works well so far. All right. So given that we can compute the true site pattern probability of any of the quartet trees, we can also compute the true site pattern probability of the quartet or quartet network or quartet, which is a 4 species with a reticulation, which is basically the sum, the weighted sum of the site pattern probabilities that are computed for each of the quartet that is decomposed from each of the display tree that has same 4 tips. So for example, true site pattern of this quartet network, we're going to call PQN, is basically the true site pattern probability of this quartet multiplied by gamma, because it has probability of gamma, and true site pattern probability of this one multiplied by 1 minus gamma. And then we sum that to approximate the true site pattern for this network quartet. All right. So now we have a way to compute the true site pattern probability for any network quartet. In order to compute the likelihood of that quartet, we need to get site pattern frequencies from the data. And we do so by focusing at a subset of sequence alignment at a time. So here in this quartet, we have species A, C, D, and E, and we're going to only focus species, the sequence that represents species A, C, D, and E. And we're going to count how many of each of these 15-site pattern occurs in this sequence alignment. More specifically, if you look at the first row, we're going to see a site pattern A for species A, C, D, and E. So this A belongs to this site pattern where 4 species shares the same nucleotide, or represented as I. In some cases, you will observe TAAT, where first and the last species shares the same nucleotide, and the second and third species the same nucleotide that is different from first and last one. And then we're going to add 1 to JIIJ, and so on. So by doing this thing from the beginning to the end of the alignment, we will fill in this vector of site pattern frequencies that we extract from the data. And by combining the site pattern, the true site pattern probabilities for each of the quartet network, that is the composite from a network, and observed frequencies that we extract from the sequence alignment, we can compute the likelihood of a quartet by using this simple formula. And then by multiplying all of the quartet likelihoods together, we can get a composite likelihood of a network, which basically approximates the full likelihood of a network. Okay. All right, so now we have, yes, 2 minutes? 2 minutes? Oh my God. 5 minutes, okay, I'll be quick. Okay, so now we have a way to compute the fit of the data on any network topology. Our next goal is to find the topology that represents the data the best, and we do so by using heuristics. So let's say this terrain represents our network space. It has a smaller hill or big hill. We're going to call big hill global optimum, smaller hill local optimum. And as we move up this altitude in this network space, the likelihood increases. So our goal is to somehow traverse this network space and eventually get to this global optimum. So this is very simple representation of what the network space actually looks like, because network space is multidimensional, which means by adding a reticulation onto a topology, the dimensionality of the network space increases. And what's more problematic is as we move up the dimensionality, the likelihood of the network that exists at the global optimum also increases. So unless we limit the number of dimension of the space to be searched, the final network will have too many reticulations. Okay, I'm going to skip because we don't have time. So one strategy is hill climbing, which is a very simple strategy. We're going to start at a certain point in the network space. We're going to traverse the network space in a way it's always going to make an upward movement. And we're going to hope when the analysis ends, that means we reach the global optimum. But this is not always the case, because depending on where you start, you might get stuck at the local optimum, which is not the highest point in the network space. So to prevent this problem, we also implemented simulated annealing algorithm, which allows downward movement at some probability, which increases the chance of finding the global optimum. But the downside of this is, depending on where you start, it can take very long time to finish the analysis. Okay, so all of these methods, all of these composite likelihood searching network space are implemented in a software package called PhyNEST, and they actually perform very well. So we simulated data using this network topology for different size of data set, and then we compared the performance of PhyLoNet and SNAG, which are 2 very popularly used network methods, and PhyNEST using hill climbing and PhyNEST using simulated annealing algorithm. So this shows the accuracy of the 4 methods. So x-axis, the data set size increases, the number of gene trees increases. Y-axis represents dissimilarity, higher the value, less the accuracy. You might see when the data set size is small, all methods kind of suffers in terms of accuracy, but PhyNEST, our method, performs quite a little bit better than SNAG or PhyLoNet. And as we increase the data set size, the performance basically gradually increases, but PhyLoNet continues to be least performing, whereas PhyNEST using simulated annealing turns out to be the best performing. So this shows the computational cost of the 4 methods. So let's ignore this purple box plot at this point. So these two plots on the bottom, so before that, x-axis again, data set size, y-axis represents the time. Higher it is, more slower it is. So this bottom two box plots represents SNAG and PhyLoNet. They're really fast. And PhyNEST using hill climbing, they're a little bit slower, but still relatively fast. But you might see, okay, this blue plot here that represents PhyNEST using simulated annealing is much, much slower than any of the methods because it searches the network space more exhaustively. But we need to say, this doesn't really tell us the true picture because we need to also consider the time to summarize the data into a set of gene trees in prior of PhyLoNet and SNAG analysis. And that time is represented in this purple box plot. So when the data set size has less than 3,000 loci, okay, it might be true that summarizing the data into gene trees and running the SNAG or PhyLoNet method will take similar time as using simulated annealing algorithm for PhyNEST. But when the data set size exceeds 5,000, the summarizing the data into a set of gene trees exceeds the time that is required for PhyNEST using simulated annealing algorithm. So what we are saying is when the data set size gets larger, PhyNEST actually gets more faster than the summary-based methods. We also conducted some empirical data set. I used monkey because I love monkeys. So these three species on the left are three species that occurs in Asia. 4 species on the right, 4 species occurs in Africa. And this Van der Poel et al. Paper detected many integration scenarios among these 7 species, but one integration or hybridization among Asian species, one integration or hybridization among the African species, and many hybridization between Asian and African species. We run PhyNEST analysis, and we assumed if there is one hybridization event among the 7 species, we have inferred one hybridization or one reticulation among the African species. So there was enough signal for of hybridization among the African species, and we increased the number of reticulation in the final network. We continue to have this reticulation among the African species, but we additionally inferred a reticulation among the Asian species, but we never observed any reticulation between Asian and African species. All right, so in summary, I hope I convinced that phylogenetic networks are good because that can represent non-tree-like evolutionary scenarios. And for biological study, you should use explicit network, not abstract networks. And hybrid detection methods can be very useful when you have genome data, but cautious use is recommended. And we present PhyNEST, which is an acronym for Phylogenetic Network Estimation using Site Patterns, a new method to infer phylogenetic network using site patterns, and it's freely available from our GitHub repository. And for now, it takes in a sequence alignment in phyLib format, and it yields level 1 phylogenetic networks. The biggest advantage of our method is it bypasses data summarization step and utilizes all phylogenetic signal in the alignment. But the downside is user must specify how many reticulation you are expecting in the final network, and it requires an outgroup sequence to root the final network. So I'd like to give special thanks to Laura Kobako and Dave Swafford, which are the co-authors of this paper. And our paper was published about a month ago, so if you're interested, please go, please find this paper and take a look at it. And thank you for all of the funding agencies and institutions that supported my research.
¿Preguntas? ¿Comentarios?
Thank you for your talk. We now are using IBPP with reticulation in our analysis. Yes, with a model to estimate reticulation. So I don't know if you compare this method with yours and try to see the efficiency or?
Yeah, so. Thank you. So thank you for the great question. So BPP is another method that uses what's called multi-species coalescent with integration model. But the biggest difference between our method and BPP is, first of all, BPP is a Bayesian software. So we didn't include in our simulation study because for Bayesian method, it is very important to setting up the correct priors, right? In order to get a good estimate. And I'm not an expert, none of us were expert in Bayesian analysis, and we were worried, oh, if we end up choosing wrong prior for the analysis, and turns out the BPP is not performing well, it might not be the right way to conduct the analysis. So all of the three methods, PhyNEST, SNAG, and PhyLoNet, they all use a very similar framework, so we thought we can objectively compare these three methods. The second thing is BPP estimates the parameters in the given network, but as far as I know, it doesn't really conduct MCMC, RJMCMC to find the best network given the data. And the reason is because it's computationally very, very heavy. So similar to BPP, there are other Bayesian methods implemented in PhyLoNet, which we can explore how different priors result in the accuracy. But those are two big reasons that we decided not to use BPP for our simulation comparisons.
Yeah, so when you talk about the composite likelihood networks, the question is if there is a specific number of species or taxons that we need to have for making, or we can use any number of species for doing a composite likelihood network.
So the question is, for the composite likelihood method, can we use any number of species, or is there a limitation in number of species? This is actually a great question, because composite likelihood is, the computational time of composite likelihood is not bounded by the number of loci. So you can have many, many loci, and the computational time will be pretty similar. But it is limited by how many sequences it has. So not number of species, how many sequences it has. So SNAG and PhyLoNet are two most computationally efficient methods that are compared in our simulation. And their limitation is about 25 sequences. So which can be translated, if you have 25 sequences, it's going to take 2 weeks at least. And for PhyNEST, if you use hill climbing method, I think we can deal with 15 species within reasonable amount of time, which is like a couple of weeks. But if you have more than 20, it's never going to end. Thank you.
That's good. Thank you.
Could you please go back to the heuristic strategies you are using to find the optimal solution? Yeah, those ones. Yeah, anyone. No, next one. Clean beam could be. Yeah. So my question is, these strategies are always starting from a starting point, only one, apparently. So have you considered implementing like kind of bootstrapping the searching so that you can start from different points of the space? Because the way it is, probably chances are that the optimal solution is being not found, you know? But it's just a matter of curiosity.
Yes. No, that's actually a great question. And this illustration, I made it this way for simplicity, okay? So it is very important to start in different places of starting topology. And how do we find a good starting topology is the first question, because we don't want to start from like here, because it's going to take a long time to get there. If we start our search from here, we're going to find the true solution, and the analysis will end quickly. So how do we find a good starting topology? One way to do it is we run, which is already reliable species tree estimation method. And some of the methods that can infer species tree from sequence data are like, there exists multiple ones, like SVD Quartz. It's going to end really quickly. It can infer very accurate species tree. That is one way to do it. But sometimes, you know, you might not have enough time. Sometimes it's going to be like, you know, too troublesome to install this SVD Quartet on your laptop and do stuff. So another way to do it is, which is also implemented in PhyNEST, is you are going to give us random topology. So let's say random topology is right here. And then if you turn on the option that is called start NNI, it's going to conduct an NNI movement which slightly modifies the starting topology. So you can start in multiple different places in your, to initiate your search. And in this case, you can't really sure if you are in a good place, but you can be sure that you are studying in many different places and hoping one of them will lead you to the global optimum.
My question is, do you think other biologists or scientists in general will use your PhyNEST to something besides hybridization? And what do you think it would be great to use it?
Right, so the question was, can PhyNEST can be applied to other biological process other than hybridization, right? So at this point, the model that is implemented in PhyNEST, we assumed hybridization, right? So we recommend to use this to infer hybridization or integration. But the previous studies have shown, even though you use some methods that are designed to infer hybridization, and you try to infer polyploidization, for example, it kind of gives a good answer. So we're not directly modeling, for example, polyploidization. So if you, okay, why does this doesn't make sense when I have polyploid species? It might not make sense, but simulation study shows, okay, it kind of makes sense, they kind of work well, but it only shows it works well. It doesn't guarantee this can be used for other processes. Yeah, some other reticulation processes can include recombination, polyploidization, or gene duplication and loss, and there are different methods that only assumes those biological processes. So if you want to infer those, you can definitely try other methods that are modeled for those processes.
Excellent. I know, I love it.
How this method deals with incomplete linear sorting? Sorry? How this method deals with incomplete linear sorting? Estimate incomplete linear sorting in some way? Yes, that is not due to the hybridization.
Right, okay. So the question was, how does PhyNEST deal with incomplete lineage sorting? And this is important thing to consider because incomplete lineage sorting and hybridization will result in very similar footprint in the genome. So one way to deal with distinguishing, or before network, how do we distinguish incomplete lineage sorting for the species tree estimation is to use a multi-species coalescent model. So multi-species coalescent model tells us for a given unrooted quartet, the probability, the topology that has highest probability is always the true topology. So we are directly using multi-species coalescent model in our methods to deal with incomplete lineage sorting and to distinguish that from hybridization, which is basically the same concept applied in HYDI for hybrid detection, and SNAC and Phylogenetic and many of those.
Hi, do your PhyLoNest have any way of parallelization or optimization? I'm thinking about large datasets or datasets that are like, have individuals that are really closely related and might take longer to, sort that.
That's right. Yeah, that's a great question. I've been keep saying like, oh, like it's gonna take long, it's gonna take weeks, but yes, we have a way to parallelize it. So one way that is implemented is depending on how many cores you have on your cluster or on your computer, you can parallelly run each of the independent searches simultaneously. So if you want to run a network space search, I parallelize this because network space search is the place where it takes most of the time. So you can parallelize different searches simultaneously, but the problem is how many independent search is enough? And by default, we set 10. So you can run 10 independent searches, and we hope we get the true network. But some of the simulation studies that we have conducted is using empirical data. When we run 50 independent runs, it only found twice the true topology when the data set size gets larger. So we have a way to parallelize this to get faster. Our next step is to parallelize, decomposing quartets and computing likelihood, which we hope is going to speed up the process.
Thank you, Kevin. Thank you very much. I think it was very enlightening for everyone, and thank you for your questions as well. Yeah. Okay.
Thank you very much.